Hi everyone, and welcome to this series of short videos devoted to CytoPipeline, another tool for building and visualizing pre-processing pipelines for flow cytometry data. In this video, I will provide a bit of background information about pre-processing steps and what it means to automate these pre-processing steps. In particular, I will also try to provide a set of typical examples and how to automate them. If you are already familiar with pre-processing pipelines in R, you might want to skip this video and jump immediately to the other ones. If not, let's get started. And so, what does it mean exactly to automate pre-processing steps? Let's zoom into some examples. And let's first have a look at the step which is called the doublets removal or singlet gate. So in a typical pre-processing pipeline for flow cytometry data, we need to eliminate a series of undesirable events from the dataset. And one type of events is doublets. So doublets are the contrary of single cells. And typically a user using manual gating, so not using automation for doing this, will do this using an interactive software and looking at the data in a 2D representation following these axes, FSCA, so the forward scatter area and the forward scatter height. The idea is that the ratio between the height and the area is to be constant if you consider only the singlets. And so the idea would be then to eliminate the events that are in this region, because there typically the area is too high compared to the height. So a human operator would end up designing such a gate, polygon gate, as an example. So if we want to automate this process using code, then we need to use a package, an R package, and a method of this package. Here I have selected singlet gate from CytoPipeline, so that's my own implementation of the algorithm. And the principle is very similar. So as the singlet events are spread around a constant FSCA ratio, if we mentally imagine the straight line going through the observed median ratio, then we can build an interval, a vertical interval, of plus or minus a specific quantity, which represents a specific number, 4, 5, 6, of standard deviations around this median ratio. And so if we do this, this would create a kind of parallelogram gate. And nmat, so here the nmat would be a parameter to be set by the user to define the width of the vertical interval. And so if we implement this in code, in fact, what we would do is implementing this kind of code chunk, where we first start with invoking, so loading the packages we will need in the subsequent code, then calling two different statements. So first calling the function read.fcs from the flowcore package, which basically reads the sample file from the disk. Store that into a variable which is flow frame. Flow frame is a typical structure we use for flow cytometry data representation. And then pass this flow frame as input into a function, our method, which basically implements the algorithm I've just explained before. So, and in this case, this is a function I called remove doublet CytoPipeline. And there we see the same. So beyond the flow frame here, we have other parameters, the two parameters specifying which are the area channels and height channels we want to use, so FSCA and FSCH here. And we find back the nmat parameters, so this is typically a parameter that the user would use to fine-tune the algorithm behind remove doublet CytoPipeline. Okay, if we zoom now to another example, next step, which would be to remove the debris. So typically, removing the debris would be done by a human operator using manual gating by looking at another representation in 2 dimensions of the data. And this representation is following these two axes, FSTA, forward scatter area, and side scatter area. The idea is to eliminate all the undesirable events that are near the origin, because those correspond to events of small size related to forward scatter and small granularity here, side scatter. So typically a user would define a gate, maybe this kind of fancy polygon gate, to isolate the good events and reject the bad events. And we will end up with the set represented on the right hand side. Now, if we want to automate this step, again, we need to have a method doing something similar. And here I've selected the package flowclust, which basically enables a specific type of clustering algorithm. And so the idea behind gating using flowclust is to fit the 2D density of this data set with a mixture, so an addition of a number of student T distributions. And here the number denoted by K has been set to 3, because here we see that we have 3 different clusters, and we want to get rid of the cluster that is near the origin. Again, this method would take as parameter the number of clusters, so here the number of student T distributions, and some additional secondary parameters, for example, the number of degrees of freedom of the student T's, and other parameters, for example, a level which specify a threshold which eliminate all events considered as outlier beyond a specific quantile. And so if you apply this algorithm here, we will obtain something that is very similar to what we have on the right hand side. Okay, so if we do this in code, and starting from what we had obtained earlier, we will add another step, which is here calling the function remove debris flowclust tmix. And again, we can see that the flow frame here has been passed in the next step as input to the remove debris step. And again, we have other parameters corresponding to the exact name of the channel that are used to do the clustering, and then the additional parameters that are used by the user to fine-tune the algorithm. So here we can see that we have a chain of flow frame going from output of a specific step to the input of the next step, and so on. Note that in this code we could have used also another notation like the pipe operator. So moving to a third example, and zooming now on the so-called quality control. And in the context of flow cytometry, quality control often refers to signal stability control, meaning that we want to check whether the signal is stationary, and we want to have a stable signal in time. So typically then we will need to have a look at the time dimension and select a channel in the y-axis and look at the stability of this distribution in time. So here what we find is that the situation is quite good. Only at the very end we have maybe a part of which distribution is a bit different. And so typically a user using interactive software to proceed with the quality control would probably use manual gating like this one. So selecting most of the events, but forgetting about the last events in time. Again, we have ways to automate this using algorithm and using packages in R. And here I've selected PicoQC. PicoQC, the reference of the package, so the reference paper is here, uses some machine learning techniques, outlier detections, isolation trees to eliminate bad quality events. And PicoQC will do that not looking at one channel at once, but using all channels at once. Note that this method comes with various parameters, and again, those parameters are used to tune the algorithm, but typically this can be sometimes difficult to select correct values, and fortunately the algorithm comes with some default values. Of course, it's not the only method that we can use for quality control, and we have other competing methods like FlowAI and FlowCat. And so if we translate the use of this PicoQC algorithm into code and piling up the new step to the already existing steps, so we will add a new chunk of code where basically we have added also compensation and scale transformation, because it, as said in the paper, PicoQC needs to run on compensated and scale transform data. So this is a kind of naive code that we will pile up in order to describe our pipeline. Now, in CytoPipeline, we will not use this naive code, but rather define the pipeline centrally to make it more easy to adapt the pipeline and to visualize the results. And that's it. In this video, we've gone through a set of typical examples of pre-processing steps, and we've highlighted what it exactly means to automate these steps in our code. In the next video, we'll talk about the dataset that will be used throughout the demonstration of CytoPipeline.